All right. Happy HIE Awareness Month, everyone. We are nearing the final 2 days to close out the month. It has been an incredible month full of education, full of community gathering. We just finished up our last of Hustle for Hope events, and what we've been trying to do through these wonderful Q&As throughout the month and throughout the year is bring topics of interest to our community. And this one, I'm so excited to bring to our community today. We have 4 absolutely amazing researchers. We talk about accelerating research and advocacy for HIE. A lot of times people, don't necessarily, get a chance to hear more directly from the researchers themselves who are piloting and moving forward this progress in the field. And we're just fortunate to have 4 exceptional people today who are have very diverse backgrounds and interests. And so I'm going to do a quick introduction so everyone knows who they are and give each person a chance to introduce themselves a little bit more about the work they're working on and kind of what piqued your interest getting involved in driving forward research for HIE. And so today we have Dr. Kenji Cunnion from Rialto Life Sciences, who is the Chief Medical Officer and a pediatric physician. We have Dr. Lina Chalak from UT Southwestern, who is the Principal Investigator of the Cool Prime Mild HIE and Cooling Study. We have Dr. Brian Kalish, who's the Director of Research and Neonatology for Sick Kids in Ontario. And we have Dr. Nikki Robertson, who is the Professor of Perinatal Neuroscience at the Elizabeth Garrett Anderson Institute for Women's Health at the University of College in London and University of Edinburgh in the UK. So welcome, everyone. I'd love to just do a roundtable of the Hollywood Squares of a little bit about each of you. So Lina, if you would just start us off.
Hi, everyone. My name is Lina Chalak. Like Betsy mentioned, my passion is the newborn brain health, and I've been doing HIE studies since pretty much as long as I can remember. Started in the 1990s on piglet translational asphyxia models, learning from Nikki and others, and then moving to clinical trials with the Neonatal Research Network, and then leading my own trials with the Cool Prime, and others. I'm a professor at UT Southwestern, and I'm the director of the Neonatal Neurology Program, the Fellowship Program, and the Chief of Neonatology. And I look forward to answer any questions that I know the answer to and to learn from you on what I don't know.
Great. Brian?
Hi, everyone. My name is Brian Kalish. I'm the Director of Research for Neonatology at the Hospital for Sick Children in Toronto. I'm a neonatologist here at Sick Kids. I'm also a scientist in the program in neuroscience and mental health. My lab has sort of diverse interests. We have a fundamental developmental neuroscience interest in fetal and neonatal neurodevelopment, particularly how the immune system and the brain develop together during early life. And then we have a translational interest in brain injury and new therapies to protect the newborn brain. And that's what brings me here today. I'm passionate about trying to develop new therapies for HIE, and I'll talk about some of those new approaches today.
Thank you. Nikki?
So good evening. Well, it's evening here. Good afternoon, everyone. So I'm Nikki Robertson. I have been interested in HIE or neonatal encephalopathy for probably 25 years, 30 years, and have been researching, starting off looking at imaging and MR spectroscopy and how to prognosticate. But then was lucky enough to have the opportunity to take over the leadership of the preclinical lab at UCL, trying to assess new therapies that can be translated into clinical effective treatments for babies. And over the last 2 decades, we've been researching multiple potential therapies, including xenon. We've done a clinical trial of babies using xenon. But the most promising in my lab anyway has been melatonin. And we are now at a stage where we're doing some early phase clinical trials of melatonin in babies with moderate to severe HIE. I'm a Scot. I don't sound Scottish, but part of my work involves running the Neonatal Neuroprotection Research Group in Edinburgh. And there we're looking at stem cell research, umbilical cord, mesenchymal stromal cells. And so I'm quite busy going up and down between North and South. I'm really happy to be here, and I look forward to hearing your questions and talking about neuroprotection.
Thank you so much. And Kenji?
Thank you, Betsy. I'm Kenji Cunnion. I'm an infectious diseases pediatrician, spent 30 years providing clinical care, also ran a basic science laboratory for over 2 decades. I am the co-inventor of a small molecule now in phase 2 clinical trials for babies with moderate to severe HIE. I am the co-founder of Rialta Life Sciences, which is the company that my co-inventor and I quit our day jobs to found in order to accelerate the development of this molecule, which will hopefully bring brighter futures for babies with HIE. Thank you.
Thank you, everyone. Great, great intros. And so, we kind of talked a little bit about, what you're doing now and, what kind of passionate about HIE. So, I'd love to start this off with what your perspective is on the importance, because we're a patient advocacy and support organization and partnering with families and advocacy organizations in the work that you do. And so we'll start with Lina.
So I started partnering with Betsy and Hope for HIE Foundation as I was trying to answer questions about how do we move the field forward when it comes to mild HIE, because people were trying to do randomized trials and we were struggling with consent rates. So those pilots were not being completed. And the studies were having to change design because nobody would agree to be in a control arm. And that bottleneck kind of needed some answers. And I realized if we as physicians cannot even agree on what's the best thing to do for the babies, it is not fair to not involve our stakeholders with us to try to find those important answers. And rather than taking no, you cannot get a trial on mild HIE and just people do what people do, we decided to organize the chaos. So Betsy and I started with a small focus group, and we learned that parents wanted to help science and understood the nuances of mild being not so mild all the time, and the neurological assessments being limited in their accuracy in the first 6 hours after birth, and were willing to help us. And the way to do it is by following regional guidelines. And so what we did is we surveyed what would be important outcomes. And we learned that the longer you follow up, the more relevant it becomes. And that function is more important than structure of an MRI or a test. And we also learn about the important side effects that matters. And we also learned from that what happens in the NICU for 3 days, the short term, parents are willing to accept that for any potential benefit in the long run. And we also learned about potential new things that we had not thought about, like the bonding and the attachment after hospital discharge. And we designed our Cool Prime trial accordingly. And therefore, I'm proud to say that this trial is the first that in HIE, there are other parents' trials that you're going to hear about from our speaker panel and from Betsy. But in HIE, that is the first stakeholder engaged trial that the effect size, the primary outcomes are all determined with the help of the family foundations. And Betsy has been tremendous ally in this. And I'm happy to report that the study has a consent rate of close to 75%, which is the highest we've ever had in mild HIE. And we're 6 months into recruitment. So for me, it's eye-opening experience. It's wonderful. And it's the only way moving forward. So there should be no studies in any field that does not involve the parents, as far as I'm concerned. I hope I answered the question, Betsy.
Thank you. Yes, I'm gonna flip it over to Brian.
Sure. So in terms of my background, so as I said, I trained as a developmental neuroscientist and then moved here to Sick Kids about 4 years ago. And a large portion of my research program has focused on basic developmental neuroscience. But since coming here to Sick Kids, we've really grown our efforts to try to develop new approaches to trying to improve the outcomes for infants with HIE, which has been a deeply rewarding experience. And I would say partnering with Betsy and Hope for HIE has been a critical part of that experience. We've been advancing two specific efforts. One is a trial for something called remote ischemic conditioning, or RIC, RIC, which is an intervention where you temporarily restrict blood flow to an extremity, and that leads to the release of chemicals and molecules that then protect the brain and other distant organs from injury. This has been widely used in adults, for instance, with stroke, but really hasn't been advanced in younger patient populations, including infants with HIE. So that's been an effort that we've advanced here and just completed a clinical trial demonstrating the safety and feasibility of these efforts. And for that study, it's an intervention that sounds scary to many parents. And I think engaging with families and understanding how to present this information in a way that's understandable, especially during the scary first few days for a family with a child with HIE when they're undergoing hypothermia is critical. And initially, we faced challenges with recruitment into the trial. And I think once we understood the benefits of engaging with families and presenting the data in a way that was amenable to for families to understand, those first few days was a key factor in our success. We just completed that trial and will be submitted for publication. And then, as Betsy mentioned, we're have just designed and are shortly going to be embarking on a study to use the drug metformin, which has been used for over 60 years for infants with HIE. I'm sorry, for infants with children with type 2 diabetes. We're looking to repurpose this drug as a neuroprotective molecule in infants with HIE, as has been done for other children with brain injury. And as part of that, we've been working with Betsy and Hope for HIE, as well as many other families, to try to design a really patient-centered trial. And I think both of these experiences have illuminated how important it is to really engage early and often to make sure that the design of these studies is meeting families where they are during this very difficult time.
Thank you. And we just had a comment come in from a family that said, Dr. Kalish saved my son's life. So that's really nice. Glad that they're watching, and always great to hear from our families who are thankful for your care. So Nikki, switching over to you.
Thank you, Betsy. So I've spent, as I've said, 20 years running a preclinical model, and have been able to take, I think, 2, maybe 3 therapies into early phase clinical trials. I think what was very clear to me from a recent C4C meeting in Rome was that actually partnering with Hope for HIE and parents is as important for the preclinical model, so that you know what's going on and you can inform us what's important and what you'd like to see. And so now that's sort of changed, and we are involving parents and advisors in our preclinical model design as well. But more than anything, I think it's been the help from parents and advocacy groups in trying to develop early phase clinical trials. And as Brian has mentioned, some of the difficulties in getting across complicated information in a time-pressured and very stressful period requires the information to be packaged in bite-size and appropriate ways. And I think for our early phase 1 dose escalation study for melatonin, it has been enormously helpful working with parents who've been through this, who understand how difficult it is to take in complex things. So I echo a lot of the same sentiments from Lina and from Brian, in that it's a wonderful journey, and we need to start from the very beginning to work with Hope for HIE and other parent advocacy programs. We can do better research that is more focused, and it's really important to know what the important outcomes for you all are. So really, I think echoing a lot of what has been said.
All right, and Kenji, and just to note, so Rialta is in this call, the representation of non-kind of academic and into your own, we talk on pharma or industry side of things, which I think is a really interesting, I'd love to get more of that, your obviously input to share it out, because I think it is a little bit different. With just the, that journey of, discovering something as a physician in a lab, and then these spin outs that are happening in many different facets. It's, I'm excited to see so much happening with HIE. So would love to hear more from you, Kenji.
Thank you so much, Betsy. Yeah, I do come from even though I'd come from an academic world, Rialta is a startup biopharma, although maybe we're not startup anymore now that we're in multiple phase 2 clinical trials. But the partnership with Hope for HIE for us goes back to 2018 when we initially founded the company. And so our organizations have really grown up together, which has been a wonderful experience. In the development of our trial, we have worked with Betsy and her team at Hope for HIE to develop tools that we felt were important for the trial that didn't exist, at least not for babies. So patient-reported outcomes is something that's very commonly done in the adult world, but there really weren't good tools for family-reported outcomes for babies. So we've been working with Betsy and her team for, my goodness, it must be almost 2 years now to develop the tool that will be trialed as one of the many aspects that we're looking at in our interventional trial. But that's really been a wonderful collaboration. Betsy drew upon the key opinion leaders that she has access to, did focus groups and trialing it, pressure testing it with families to refine the tool and make it much better than what we initially started with. So we'll see how that performs and refine it even further. And so, you're probably familiar with NASA saying, "Oh, well, we, through the work that NASA has done, we develop components that are now in mobile phones and in robotics and all these other things." So this is sort of like that. Like that's not our primary mission, but through this collaboration, we're sort of developing offshoot tools that will hopefully be helpful for everyone. So the combination or the collaboration with industry, which is where I currently am, and advocacy groups is something that has not been as robust as it should be. Traditionally, there's been concerns of cross-contaminating the good advocacy groups with the evil pharma companies, but we should be working together for the benefit of all our patients, and because we can do better together than we could ever do individually. And families are absolutely critically important, particularly in situations like the condition of HIE, where there is no approved pharmacologic medicinal intervention. There's cooling, and that's it. As compounds, perhaps ours, perhaps one of the compounds some of my panelists are working with, get closer to potential approval by regulatory agencies, the input of families will be absolutely critical. The FDA sounds like sort of a scary, huge government agency, and they are a huge government agency. But we've been having a dialogue with them around HIE for over 5 years now, and sharing ideas and how do we get to something that will help the lives of babies. And so this type of collaboration can be done, should be done. But these types of regulatory agencies like the FDA, they need to hear from families, because that's part of their federal mandate, is to improve the health of the citizens of our country, obviously regulatory agencies for other countries. But if they don't hear from the families, then they sort of start to default to this safety, 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 and they get super duper cautious, and they become completely risk averse. So the voices of families to help bring new therapies forward is absolutely critical, particularly for regulatory agencies like the FDA. And I'll stop there.
Thank you. And, I want to kind of dovetail off of that a little bit to share that we have been also working a lot with the FDA and the EMA in Europe, and also regulatory agencies in other parts of the world, as these, events, for these stakeholder events are happening, as different things are moving through the pipeline, and as there's educational events that like the regulatory agencies themselves host. So, neonatal clinical trials just are tough. Everyone wants to protect very fragile babies. And I think very, clearly, as a parent myself, whose child went through cooling in 2012, which was kind of at that middle point of the cooling journey of research, where the 2 weeks after we got home from the NICU, on the front page of the Detroit News was the long-term study results of the whole body cooling trial through NRN in the US. And then we have families in Hope for HIE who have participated in the original Toby trials in the UK, and in other trials worldwide with HIE. Probably nearly every trial we have representation of from a family. And I think about the complexity of cooling and how families, we all talked about this like narrow window of, of cooling and things like that we know is so critically important and is so tough as well. It makes it, the stakes are pretty high with this, with getting intervention and all of that. But we found ways to make that happen. 6 hours is a very tight time to consult and start a therapy. We talk about this with neonatal seizures and with all sorts of other things that are part of the neonatal landscape. And when timing is so critically important, I think of the families that were approached for these trials. And of course, there was nothing before cooling. So, I think that was probably the call to action of counseling families was, we're hoping we'll give your child the best outcome that we can give them. But cooling sounds like science fiction. And, so I love to, obviously it's become standard of care in many parts of the world, from moderate to severe HIE. Lina and team are expanding it for mild to test that. And so many things we're learning about, even trials that don't show, a positive improvement like we had hoped, like the HEAL study. We had so many families in that, but now we have these secondary analyses that are coming out that are helping us accelerate and move forward. So I want to clarify for families too that are tuning in. We have a lot of families popping in and out and letting us know that they're watching as well, that when we talk about this pipeline of science and looking at how things are developed, and going through the big question of, is this safe and is this effective, right? And we're looking at a really fragile population. It's the brain, so it's the core organ to who we are and who, what makes us able to be in this world. And it's so complex, and you all are taking on such a complex task with trying to accelerate this forward. But, I've had a wonderful opportunity the past couple of years in particular to learn more about this preclinical work. And when we talk about preclinical, that means not in humans. So that is like the animal model work. And there's lots of different steps that, the regulatory agencies look for and data to look. It's a lot of work. And as Nikki mentioned, like years and years of work to get to different points of this. In particular, we have smaller, animal models, mice and rats and things like that. And then we have larger with pigs and sheep and, primates and all sorts of things. And, there's good reasons why we go through these checks and balances to do all that preclinical work. Because we do need to make sure that this can flow through, and there's going to be a risk benefit of many different things and how we evaluate those. So I just wanted to clarify with families when we talk about preclinical and then working through this, this pipeline, the clinical trial part is in humans, and that even has to go through a lot of layers before you get to babies. So typically you have to look at how something, results in healthy adults, and then looking at potentially, unhealthy adults, a population, and then, maybe pediatrics, older kids or younger kids, before you get to neonates. So there is quite a complex process. And also to say, and just because I know that this comes up often, we're talking a lot about neonatal HIE today, but there are lots of other work that we've been highlighting and will continue to highlight, of the many ways that HIE impacts and the ways that there's a ton of research going on for that as well. So I don't want everyone to think, my kid missed out on these things that are happening. Mine too. But, this is how we learn more, and hopefully it's a lot of these different things that we're trying now for and hoping to move forward with neonates and babies will, maybe some will unlock a code for pediatric, or there's other things out there already that are being worked on. So I just wanted to share that with our families as they are listening to this. So switching gears a little bit, we're going to focus on the two of you that have active trials right now that are enrolling. And so I know, Lina, you talked a little bit, and Kenji both, about your studies, but I'd love to kind of go into more of the mechanics of why we heard a little bit of the why of why this is moving forward to look at the science, and what have you learned so far as you're enrolling patients and going through this project. So Lina, let's start.
Yes. The first thing we learned was that obtaining the consent in the first 6 hours is tough for families. They're recovering, they've got sedation, they've got families trying to find their other children. Mom and dad are not in the same place. So what we learned is, because the site is the one that is initiating the treatment, and we have some sites in our grant where the standard of care is to do hypothermia for the babies with mild HIE. And then we have some sites where the standard of care is to do normothermia, meaning you avoid the increases in temperature, which we know are dangerous, but those sites feel we don't have enough evidence to adopt hypothermia in clinical practice, because the knowledge and the evidence collected to date definitively tells us that mild is not so mild. So we know that there are outcomes that we need to follow and trajectory of outcomes, but none of the studies out there are conclusive to tell us that hypothermia works and can change those outcomes the same way it does for babies with moderate and severe. And one could ask, well, why does it matter? Why don't we just do it? The truth is, time zero in animal studies is very clear and identified. Time zero in babies is a complicated business. You're lucky if time zero is around the time of birth, because these are the babies that respond best to hypothermia. But some babies that we so-called label mild could be babies who have had an older insult, acute on chronic, or a more severe insult that partly resolved, and now you're seeing the tail end of it and you're calling it mild. And for those babies, time zero is not at birth and hypothermia might not work. So until we compare head to head the practice of normothermia and hypothermia in babies with mild HIE, we do not know the reality of how effective it is for that group of patients. So the study allowed us to move forward, and now finally we can recruit, because the sites that are cooling are having the parents say, yes, please, I want to help with following the trajectory of the outcomes of my babies, and I want to help other babies. And at the sites that don't cool, they said, okay, if we're not providing hypothermia, at least let's get a trajectory of follow-up outcomes. And if there are any problems at any time point, then we could do early referral intervention. So the knowledge, the shared knowledge pool has increased because of that practice. The parents are consenting to it. They like that study design because they got to vote on it. And then they also guided us about when to ask for consent. Since hypothermia is being applied based on the site practice, so some sites are randomized to cooling, some sites are not cooling based on their practice, then there is no pressure to get a consent within 6 hours when mom is still recovering, dad is trying to go see his other children, and there's so many factors that confound the situation. And so while we initiate cooling or normothermia within the 6 hours, we allow up to 24, and even we give them a margin of 36 hours to get the consent, so that the parents would be able to have thought about it, discussed it among themselves, and make a better informed decision. So we've learned how and when to ask for consent, and we have learned what are the outcomes that matter. And we have so far recruited over close to 50 babies in less than 6 months, with a consent rate of 75%. So we are very excited. And 2 years from now, we're going to be able to know, is this a small effect size? Is this a big effect size? Is this harmful? Or do we need to do more studies? This is very exciting. This collaboration has been wonderful. We have 15 academic sites that have NeuroNICU within the US, and we have one site in Ireland that is helping find the knowledge and the information move forward. And we have lots of secondary studies planned that we're going to start to work on, from EEGs to economic analysis to school age outcomes. And we're just very excited that there is no more bottleneck, and we're able to try harder to find answers, because we owe it to our community and to our families.
And Lina, I just, if you could talk a little bit about what the protocol is of how you're identifying these mild babies. And what I think one of the things that I have been so struck by is with the EEG, and kind of talk through a little bit of that.
Yes, thank you. Excellent point. So it helps to have our monthly meetings, because then we can be fresh with ideas. Thank you, Betsy, for reminding me. So first of all, for any of you who are interested to learn more, we have a website that's active that people can visit and learn more from. So if you go on www.coolprime.org, you can read about the details of the study, so to speak. And see how far we are with the progress and the enrollment. There is the site that is open to the public, and then there is the site where we do our confidential privilege patient information and forms and things like that. Within 6 hours, the providers would be looking at each hospital, at each site, at whether the baby has a buildup of acid in the blood. The same as prior trials. If you have an acid buildup in the blood, then you need to have a neurological assessment. and called the modified SARNAT score. And we have a form that's validated exactly for the study. And what we do is we like to know that baby has an acute event around the time of birth, because again, those are the babies that are going to respond the most. If the baby has not been moving for 5 days before the birth, then those babies are less likely to respond to treatment. So acid buildup, something acute around the time of birth, you get the attending to do a detailed examination. If your baby has more than 2 abnormalities on exam on that form, as long as they're not 3 moderate or severe, because those babies, then we need to immediately cool them. And if your baby has seizure, whether clinically or on EEG in that first 6 hours, we need to cool immediately. There is that we know helps, and that is not mild. But what we uncovered with the protocol is, because we're asking the sites to put an EEG or an amplitude EEG on the baby as soon as possible. What we started uncovering is there's some babies that are in the study, their exam is mild, their EEG first is okay, and then after 6 hours, usually somewhere around that 12 to 18 hours, there is evidence of electrographic seizures. And for those babies, when we see seizures, they're still in the study, they're going to be analyzed separately, but we allow them to be cooled, obviously, because there is evidence, albeit not perfect, that shows that if you identify those babies to have seizure after HIE in that first 24 hours of life, the effect is not as strong and as beautiful as when you identify seizure in the first 6 hours and cool, but it's better than no effect at all. So we have already discovered that just by the simple intervention of putting an EEG on the baby early on and tracking it that first 24 hours, we've discovered that we have about 25% of the babies that are having electrographic seizures, that we're treating the seizure and we're allowing those babies to be cooled. We wouldn't have found that information if we did not have the EEG on. And those babies, a lot of them, the majority, 2/3, were not having any clinical manifestation of the seizures. We know how baby seizures are subtle, and they don't always need to manifest like a marching tonic-clonic seizure. They're just on the EEG. And just that part has already helped me move the field forward and get that information out there. We were not putting EEG on all of those babies. We were so-called thinking they are mild. And now we're discovering, not so mild. So very useful, just as part of collecting data in the hospital. We're even recommending MRI. It's not mandated, but recommended, especially if you have seizures and if you're cooling those babies. And we're doing a trajectory of discharge exam, 3 to 4 month surveys, 1 year exam and 2 year exams, so we could follow the trajectory of those outcomes.
Thank you for sharing that. I just thought that was like such a poignant thing of, it's not standard of care. It's not, because, you want to do less interventions, and EEG right now, as we know it, has so much, that's going on, and it's overwhelming for multiple reasons. So I just thought it's so fascinating to learn about. And, we know, every day I have families that reach out to us that are, have older kids and say, my friend told me about your organization. Here's my story. Do you think it was missed HIE? And we just didn't have, the focus on mild really just has not been as equitable as moderate to severe. So, answering these questions through this research is so important to drive forward a better understanding for sure. Thank you for sharing that. So Kenji, I'm going to move it to you. So what are some key takeaways so far of the STAR study?
So we've started enrolling last summer. So it's a phase 2 interventional trial. It's our compound is a small peptide, which means like it's a tiny fragment of a protein. It modulates aspects of inflammation that no current medicines can do. And so that's why we quit our day jobs to found this company, is because we felt this was a unique opportunity to put a new tool in the hands of physicians to treat conditions like HIE, where we don't have good tools currently. I'm sure my panelists would echo the sentiment that some of the most frustrating parts of our job is when we can make a diagnosis. We know what's driving the underlying disease process, and we know we don't have the right tool for it. Sort of the classic situation for inflammatory diseases is steroids. So you know the saying, when all you have is a hammer, everything looks like a nail. Well, everything that falls under the umbrella of inflammation, physicians hit with the hammer of steroids, even when it doesn't really make all that much sense from a mechanism of action, what part of inflammation is involved. So this is a unique tool. We've been recruiting since last summer. It's on top of cooling for moderate to severe babies. The trial has been going very, very well. The safety, it is a placebo-controlled trial, so we are blinded, but the safety looks fantastic so far. We know the drug levels were exactly where we wanted them to be. So in terms of both safety and the likelihood to be able to produce an efficacy signal, we are incredibly grateful to all of the families that have agreed to participate. This is really the only path forward towards eventual FDA approval, regulatory approval of a new medicine. It will, one of us will eventually get to the approval of the first ever medicine for HIE. And it opens the door for a lot of other things. So in the United States, there are approximately 400 cooling centers. There is a country just to the south of us with a birth cohort of 2 million babies a year where there's no cooling available. So there are lots, and the rates of HIE are 3 to 5 times higher in the developing world than in the developed Western world. There is lots and lots of need out there for a drug because cooling is not accessible for the vast majority of the infants born on this planet. Not only is it this a drug for HIE important for the babies that cannot have access to cooling, but it also opens the opportunity to potentially have a tool for other inflammatory disease processes that is injuring or harming and killing babies, even premature babies. So being able to get approval in a term population like this provides opportunities to potentially positively impact other disease processes, again, where we don't have the right tool available at this time to improve the lives of babies and impact, the entire curve and trajectory of the rest of their life. So we have a lot. We dream big, but why bother if you're not going to dream big? So I'll stop there. Is there anything in particular you'd like me to address, Betsy?
I just want to say that we know that it's so complicated with all these factors with HIE, right? And then to ask someone, the perception of why education and literacy, health literacy building in that really short period of time is so important. Because it is something that is new, right? It's not metformin or melatonin or cooling that we already know and are testing in a new cohort. And it's just like escalating challenge after challenge. But I think that's where this partnership is so great to come together and to hear from families of what really helps them or would have helped them in that period of time to help understand and make an informed decision about it. Because these, and really trying to advocate too, because these consent forms for everyone are insane. They're 20, 30, 50 pages. And how do you distill this concept in a short period of time to help people understand what they may sign on for. So it's, I love tackling difficult problems as well. And I think this teed up really nicely Nikki, who has a huge heart and passion for low and middle income countries as well, because that is, I know a ton of the work that she's been doing is looking at how we can resource in under-resourced countries to make a big difference. So I'm so glad that you brought that up, Kenji, of talking about that cooling doesn't work in all settings and is not feasible. And so, Nikki, let's talk about upcoming trials and what your plans are for these, and how and you can definitely speak a little bit more to the passion and the problem of the inequity around the world as well. I know that's such a huge part of your care too.
Thank you, Betsy. And I just wanted to say that there is room and a very big need to develop a cocktail of therapies so that we can make sure that we have at least one or two that are effective, but also to treat the different phases of the HIE evolution, because you've got different pathogenic mechanisms and brain injury mechanisms happening at different times. So we need to think about not just the acute phase, but the subacute and more chronic phase and therapies that can help then. So I'm very passionate about I was born in Africa, and I've done quite a lot of work in Africa about developing therapies, monotherapies without cooling for low and middle income countries. But I'm going to actually talk about the Acumen study, which is the acute high-dose melatonin study that we are starting, and that is a high-income setting trial, but we are in active discussions with the Global Network and other centers in sub-Saharan Africa to use it in one of the monotherapy trials that are upcoming. But for now, I wanted to share with you some of the work we've been doing in preparation for the Acumen study. Melatonin, you probably all know, it is something that we all produce in the pineal gland, and in tiny amounts, slightly higher amounts at around 4 in the morning, and it helps entrain the circadian rhythm. But what we know is if you give melatonin in very high doses, so 10,000 times what we secrete at nighttime, it is actually very protective. It's like a mitochondrial tonic. It helps the cells produce energy, and it stops the damage that occurs from the free radicals that are produced from too much from the injury. So we've spent 15 years working on melatonin, an intravenous form of melatonin, in the preclinical piglet lab to try and work out what dose you need, when it needs to be given. So working out the therapeutic window, which actually is similar to cooling. So we know the dose that we think we need to give, and we know it needs to be given within 6 hours of birth. But in order to take this into trials with babies, we have to start at a quarter of the dose that we think we need, and make sure that there's no adverse effects at each of these dose levels until we can reach the dose that we think is the therapeutic level. I did mention that we need a cocktail of therapies. The other key contender, and I know many of you will have read about this, are stem cells. And I think stem cells are incredibly exciting and have a huge potential to really improve outcomes because of their ability to regenerate and repair, but also to have a sense of what the brain milieu is and react to that. So they're sort of sensing and treating the particular problem. So I've spent 4 years now in Scotland using stem cells that have been taken from the Wharton's jelly of the umbilical cord from 8 donors, and these cells are then grown and processed and sent up to Scotland. They are allogeneic, which means you don't need to cross-match them. They can be given, they don't have an immune reaction. And we've been giving these cells through the nose. And what we know is that the cells migrate to the area of injury and have seen some quite exciting potential benefits of these cells with cooling. And so what I am hoping very much is that we can then move on with these cells and do a safety study in a small number of babies in the UK. And then after that, perhaps move to a clinical trial. And my last thing I wanted to say is that there seems to be a synergy between early melatonin and subsequent stem cells, in that the melatonin provides a sort of tonic for the stem cells to ramp up their effectiveness, and that may be a good combination. And that's something that we're going to investigate in the preclinical model to see if that works. So thank you.
So helpful to understand. I know Kenji said he has to go at the top of the hour. So just in case you see him pop off, thank you, Kenji, for your time today. And hopefully everyone can stick around a couple more minutes, because I know we're going to hear from Brian. But Nikki, I just want to say thank you so much for clarifying as well that, yes, there is, the complexity of HIE. It is not a one size, one medication, one dose fix all. And, I've spoken to researchers who are studying sex differences with HIE. I mean, the sky is the limit of opportunity to optimize and take care of these babies. And so, it really is our intention of bringing everyone around this table of a collaboration in the ecosystem of research to learn from one another and get excited about all the great work that everyone's doing. Because it's, again, today, as it stands, we have cooling that works for some babies, and that's it. So, we're overdue for a lot, and I'm just so excited that we get to share all of this with our community. So on that note, I'm going to turn it over to Brian, who's going to talk about timing as well for some of these that could be potentially outside of the neonatal period too.
Thanks, Betsy. I'm going to pick up a little bit on the theme that Nikki started about timing and different phases of injury, because I think that's a great point to make here that, I imagine all of us on this call would say that there's no single silver bullet to improve outcome after HIE, and that likely what we're working towards in an ideal future is a combination of interventions that are precisely timed, that are going to work together to give the best outcome in these children. And so, one of my interests is not just in the early acute phase after brain injury, but in the chronic, more rehabilitative phase after HIE, and thinking about how we can accelerate recovery and promote neurorehabilitation in infants with HIE. And while in the lab, we're certainly working on many preclinical interventions in that realm. One of the more translatable interventions is the drug metformin that I mentioned. And it may seem a bit commonplace because metformin is so widely used for type 2 diabetes. It's been given to millions of adults and hundreds of thousands of children for diabetes. But more recently, it's really emerged as a very promising neurotherapeutic. And in preclinical models of brain injury, it mobilizes the production of neural stem cells and promotes plasticity. And that's what really garnered my interest. In clinical trials of older children with brain injury, for instance, children who have received cranial radiation for brain tumors that can cause significant injury, the administration of metformin has promoted quite significant benefit. And so that's what prompted my interest in thinking about the HIE population, where after therapeutic hypothermia, we have this prolonged period of incredible potential plasticity that is currently sort of untapped. When I meet with families with HIE and counsel them after we get the MRI, I often, regardless of the finding of the MRI, I usually say that one of the greatest determinants of ultimate neurodevelopmental outcome is the home environment and the family. To me, that actually reflects the plasticity of the newborn brain. And that's an opportunity for us to promote and accelerate that plasticity. So with metformin, we've done a lot of work to develop the appropriate dose to give to babies and modeling the dose in young infants. And we're looking to do a safety and feasibility trial in young children to try and promote accelerated recovery and neural stem cell mobilization after this injury and the weeks to months after injury. I think that's what makes this unique, is that it's not something to give concomitantly with cooling, but something to give much later during the recovery phase. So I think that's a model to think about, adjuncts to cooling early on, in the mid phase, and later, each of which can address these different compartments for infants with HIE.
It's so exciting. And I know that, this is when we talk about this, pipeline and timeline of events and things like that. It's just so great to hear and share with our community that you're doing this work and that all of you, have you're dedicating your careers and your lives to accelerating this. And I'm always incredibly humbled and blown away when I get to learn more about all of the work and work with you and have our community work together as we accelerate together. Again, the energy and momentum is just something that we've seen grow in so many ways. And I'm so grateful for all of you taking the time today to share this work with our community. And so I want to close with an opportunity to give any final thoughts and any, of your favorite resources or things that you might want to share with families. And no, you don't have to say Hope for HIE, but but just that, in your, I don't know, work and things like that, of what your biggest takeaways might be that you want to share with families.
I just want to say that it was lovely to chat and to be caught up on all the exciting trials. For the families, I would say never be afraid to ask what you don't understand, what you struggle with. Our job is to help find the answers, or at least tell you honestly when we do not know. My last take home that I'd like you to remember is lots of exciting work going on. The future is bright. There is hope. If somebody tells you your child will never do that, just never use never is rule number one. Every baby's different. There is hope. There is exciting trials. Ask when you're not sure. Mild is not so mild. It deserves a follow-up, if anything. What works in a particular patient population or disease severity or context might not work in others. So it's not, hypothermia is not a one therapy fit all. And we're excited about what the future has to bring.
Thank you.
Maybe I'll go next after Lina and just say that, I think as a clinician, HIE is a challenging condition sometimes to care for because it's so unexpected. Families, in the vast majority of cases, don't expect to be admitted to the NICU and to be facing these challenges. And as a clinician, that makes it deeply difficult to try to elicit trust almost instantaneously within the first few hours after a child is born in an unexpected circumstance, and garner a family's trust in the care that we're providing. And for me, that's a very welcome challenge and something that I really cherish. And I think caring for these families is really one of the great privileges that I have as a neonatologist. And I hope that in my lifetime as a researcher, we can identify some new interventions to try to provide additional benefit. So thanks, Betsy, for this opportunity.
Thank you. And Nikki.
So Betsy, it's been really wonderful to have this opportunity. I think I echo a lot of the future is bright. I very much do hope and believe we will find therapies that are beneficial, because we know so much more about HIE than we did even when the cooling trials were going on. I think every baby with HIE is so different, and we learn so much from working with parents. and understanding the evolution of the baby's childhood abilities, and just I think the whole discipline is so much better where we're working together in partnership with parents and advocacy groups. So thank you. It's great to work together.
Well, thank you to our wonderful panelists, and thank you to everyone who has been participating in this. This will, like all of our other Q&As, become an evergreen resource. It will be on our social channels. It will be on our YouTube, Facebook, and we will also create, as usual, a key takeaway blog. And if basically the Cliff Notes version of this for everyone to come back, and if you aren't able to watch the whole thing, in one sitting. But again, thank you all for a really robust discussion today. I mean, so exciting to hear from all of you and the different facets that you're working on with this. And thank you again to our community for in, and we will hopefully get in all of you and when we can of some exciting new data. So all right, thank you
Thank you for hosting us.
Thank you, Betsy.